It's a pleasure to introduce Rob Rambo and Jeremy.
I'm a student, PhD student.
Your last name?
Hi.
Yeah, they're from the UK, from Diamond Synchrotron, and Rob was our scientist at the Advanced Light Source for at least 10 years. We worked together.
Yeah.
And, he decided to move on and became a director of Diamond soon.
No.
Yeah. Besides expert in solution scattering, he's expert in other beautiful pictures. And I'm looking forward to see your presentation, Jeremy, as well.
Okay, so thank you. I don't do SAxS beamlines anymore. Yeah, promoted to admin, so I do a lot of Excel sheets. Anyways, this work is kind of inspired by the last couple of years in AlphaFold and all that stuff. And it's a collaboration with Chris Pryor, who's up at Durham. He's a maths professor there, and John Essex, who's into molecular dynamics at Southampton. The PBS of principal beamline scientists, the SAxS beamline, is Nathan Cowson, and John Tanner is also a big part of this talk that I put together about a year ago. So I think when we go forward, you know, this was kind of the view of SAxS for quite a few years, right? It was you got data, make a little shape, but then you really got to think about how informative is this shape, right? You know, the world view of biology as we know it is really through sequence space, right? We think about our proteins in terms of residues, those residues that are conserved. We look at RNA in the same way, and likewise DNA. So what we really want to think about is, you know, when we started doing cryo-EM, you know, this is probably back around like late 1990s, blobology, and then this revolution in cryo-EM led to your ability to place residues, and that's where you kind of restore that sequence information, and you can relate sequence to structure, right? And in this case, you know, every residue is perfectly determined by the observation. So I think, like, if you have the starting atomic structure, like through an AI model or homology modeling, your efforts should really be placed on keeping that sequence to structure mapping, and that allows you to do, you know, your testable hypotheses and so forth. So what ends up happening is, you know, you had RosettaFold and AlphaFold came out. I was working on this protein called P65 for quite a few years. We collected a ton of SAxS data on it, and you're going, oh, this is great. Let's see what we get out of it if we use these two programs. And the top three structures from RosettaFold look like that. The AlphaFold looks like this, but you know, the Diamond shape reconstruction looks like that over there. So nothing quite fit in, and really it was just, you know, why the AI outputs allow us to maintain the sequence to structure relationship. You know, how do you kind of progress a structure that you get as a starting model to kind of fit the SAxS data, right? So what you really think about is repacking the structure using the outputs there as a starting model. And this kind of shows you some other examples. This was the fit that we used, Foxes, right? What's the fit here? AIP was another protein that we worked on. This is the AlphaFold prediction, and then that was another one, SmartCal one that we did here at SIBYLS many years ago. Structure's still not determined, but this is what the AlphaFold gives you. And so, like, if you think about like this, you might be close, right? And this discrepancy here is kind of telling you just to repack, maybe some local repacking. Here, the overall geometry is off. There's got to be some major structural change since the RG has the largest disagreements. And, you know, when you think about why you need to repack this, it's just that I think, you know, AI predictions, you know, the concept is that it tends to the mean. So if you have a conformation where a loop might be in an open state, and the other version you have in the training is in a closed state, the prediction kind of puts it in the middle here. So here are the two states that it was trained on. And so you get something kind of in the middle. So it wouldn't be, the structure you get still needs to be validated. And I think the best way to validate that is through SAxS. And then if it doesn't agree, then your options are to do some kind of MD or refold the structure. Oops, it's the wrong one. Okay, so back in the day when I was doing P65, I actually took a guess myself looking at the SAxS data, looking at the P of R distribution, knowing that there were pieces that were related to homology models, and kind of stitched together my own version of what I thought P65 looked like. I did some rounds of simulated annealing, and I could get a really good agreement with the SAxS data, right? So this was actually not AI driven. This was done nearly 10 years ago. And then, I think I did a better job than the AlphaFold here.
So simulated, and what were you using as, you weren't refining this against any, were you just sort of doing a minimization?
Yeah. So you could see like some parts of these secondary structures poorly defined, right? Like this part here is from the homology model. So what I could do is I could place constraints on Dmax, right? I knew what the maximum dimension was. I also had constraints between the domains, again, from the P of R to kind of guesstimate how far they.
And so you did this in CNS?
Yeah, I just did this in CNS. Yeah. So I repeatedly kind of did these annealing, simulated annealing refinements. And then towards the end there, I put constraints on parts that I thought were helical, right? So then it started forming these helices. Then what you're seeing here is an overlay of a bunch of different answers. Okay, this is just, again, show you the AlphaFold prediction, and then me doing a similar attempt with homology models. And finally, if you take something like this as your starting AlphaFold model, we looked at a few examples where if you just kind of do MD for a long time, do you get to the right answer, right? Do you get close to it? And long time is like 3 months, right? We did some simulations, and we weren't seeing the convergence of the structure or the sampling of that conformational space that was consistent with the SAxS data. And so the concept now is we need a way to resample the chain trace so it can provide this better agreement with the SAxS data. And so my collaborator, Durham, he's into knot theory, I think studies magnetism and things like that. He has, a different way of looking at it, knots, so to speak. So he thought about reparameterizing the topology of the protein to look something like this. I'll explain how this happens. And you can see like this is the way that he looks at the protein, but this is the actual C alpha trace right here in red. And so what we do is if you go along the backbone and you, for every 3 C alphas, right, the question you ask is what's the largest sphere what's the sphere that you can make that will touch all 3 points, right? And that the radius of that sphere is related to curvature. It's the inverse of the radius. And likewise, those 3 points will form an angle, and we can we call it, angle, like a torsion angle. And so we do this for all the known, structures in the PDB, and you can get distribution plot. So for alpha helices, you can see torsion and then curvature. Alpha helices tend to reside around here. Sheets are kind of distributed in these areas, and then linkers kind of occupy much larger space. So what we do is we can resample from this. So we kind of preserve secondary structure, and we want to resample linkers. And we can kind of do that quickly because it's a smaller computational space. And the heuristic is essentially, are we making changes to the structure that improve the SAxS model agreement, right? So the SAxS data is embedded in this the entire time. And this shows you an example. You start off where we provide a secondary structure prediction. We create a random sample of the chain based on that version of the Ramachandra plot. And this would be an example where you see here's a helix coming around. Here's another helix. This is probably a sheet. So you go around, and then now this is the initial model data agreement. So we're going to resample this, and we get this convergence to a compact shape, and then that's the final model data agreement. So this is very coarse grained.
So the first structure is initial structure.
That's the initial structure.
That's right.
That's the initial structure.
AlphaFold or anything like that.
This was a random shape.
Oh, just random.
Well, not completely random, you're preserving secondary structure.
Yeah, so it's randomly drawn from a secondary structure assignment that you make on the chip.
Oh, you forget that one. Okay.
Yeah. Okay. So, anyways, this was telling us that we could use the SAxS data, start from here, get some kind of globular destruction, right? But the output of that is only C alpha positions, right? So you get a bunch of dots, right? Beads, and then you connect them. You know the connectivity. So the next problem for us is how do you add back the residue information, right? So you can do that with modeler, which is expensive for us because we have to pay a licensing fee for everything at Diamond. So we came up with our version of adding back the chain information. And so what I did here was the Richardsons, they have this database of 8,000 high resolution structures. I ran stride on everything, which told me the secondary structure assignments for every amino acid. And then I went through every single structure assigned for a given amino acid, say it's arginine. I looked at all permutations of, you have asparagine and aspartic acid on this side that fit this secondary structure assignment. I take all known examples of that. I then align them all, and then kind of put them in order of how similar they are. So this would be an example of some amino acid here, tryptophan probably, and you can see, everything's aligned here. So you're seeing kind of the movement of that residue side chain, and then this is how the backbone itself is affecting the next residue. You can make a tree, right? So in this case, with glutamic acid, aspartic acid, valine, that fits within this right-handed alpha helix. This is arranging, by how similar they are in shape. You get a tree. So if you start from this point in the tree and then do a random walk through it, you can kind of.
So each node on this tree represents one of your triplets.
One triplet in an observed conformation.
I got it.
Yeah. Okay. And so
hold on. So this would, all the nodes here would be triplets that are in right-handed alpha, right-handed alpha, right-handed alpha.
Yeah, belonging to that.
Got it.
Yeah.
But you could do glue, asp, val in sheet, and that tree would look different.
Yeah.
Okay.
Yeah. Yeah. So then if you kind of do a random walk through it, it just it looks like it's going like an MD. Makes fun pictures. But anyways, that's not the point of this program. That's another one. So the point is like if I'm given a secondary structure assignment and the sequence, then I can sample from that to build out some chain trace. This is an easy example. It's small, but if you get much larger than that, then you end up with a lot of overlaps and collisions.
Well, all of them are concerned. How precise is the secondary structure assignment with current whatever software you use? How precise it is?
It's probably 85%. It's in that neighborhood. Yeah.
For the helix, but for turn or linkers.
I would say it's a lot lower with turns, but we tend to ignore that.
Okay.
Yeah, we just call it a linker. It's too specific. So anyways, so you take your output right? Your C alpha output. You connect the chains, right? And then we use that program to build back the location of the amino acids, right? With the all the residues there, constrained by the secondary structure assignment, and then you get this as your output, so you can see the helices are, it looks like a protein, right? And then this is the actual crystal structure of lysozyme. This is lysozyme. So I would tell you, I think we got really close to the actual structure.
There's no AI.
There's no AI.
Secondary structure prediction and SAxS.
Yeah, right. The AI would be the secondary structure prediction, right? So if you had,
Okay,
yeah.
so it's embedded as for defining the secondary structure prediction is using,
You could use AI, but
you don't use it, using the what they call that, right? No,
I think the nomenclature is DSSP.
DSPP.
Yes, DSS, that's sort of
Yeah.
like the industry standard.
Yeah, that's
Got it.
right. But it's just like we have this way now of kind of modeling or reshaping the chain, right, against the SAxS data, and then we could build back something at the very end. But you wouldn't stop here, right? You would actually take this model and then do some kind of MD simulation on it, waters, and just to kind of make it a little bit more believable. So anyways, this was the example we had where we took SmartCal1, resampled the chain, right? So we converted it to C alpha. So this is the AlphaFold output. Resampled against the SAxS data, and then we got, we think it's a close structure, and then we do the GROMACS refinement on that, right? So you start with this, and you end up with that. This is still incorrect a bit, but I think we're a bit closer to what we think the structure is. And Jeremy is going to give you a little demo. So we use, right now, it's very alpha, right? Alpha. But it's we manage this through Jupyter Notebooks, which I'm sure a lot of people are familiar with.
But before Jeremy go, I have a question.
Yeah.
So when you have the AlphaFold structure prediction anyway, and you can take the assignment of the helices and beta sheets and loops from the AlphaFold structure. That is already
That's what
Okay.
they're doing.
you don't, but you can also go straight from sequence.
Good, yeah.
That's kind of complete the ab initio from sequence, assign the sequence to the helix.
Yeah, it
You
takes
know,
forever right now.
I know, but it's pretty cool that you don't need the AlphaFold.
Well, the small protein, like the 14 kilodalton,
Really possible.
yeah, you could do it in like, I don't know, a day or two. You get up to something the size of BSA, you're looking at,
Gotcha.
it's not optimized.
Can I ask a question? Have you looked at fold seek and whether or not you can sort of take advantage of fold seek type information?
No. We haven't kind of gotten to that part yet. Yeah.
Yeah.
Somebody won't let us publish it.
Includes, they're going after the environment of the C beta, I think. And they have they've taken 20 amino acids and converted them into environments, almost like 20, but they're, but they are able to do, this is for looking for homology, homologous structures, sort of like Dolly, but much faster. But I'm wondering if that could be combined, that idea could be sort of combined with what you're doing.
Yeah, so the issue is your secondary structure assignment, right? We're kind of tied to that, and if your assignment is incorrect in some places, then you won't be able to refold it. So what we need to do, I think, next down the line is have some kind of feedback on it where, I've wasted a couple of days now. What do I need to change next, right? So, and that's where you have to go, hey, can you assess this part of the structure is weakly defined, poorly defined, or not, right?
You can move that, sorry.
Yeah, go ahead.
You can move that poorly defined of low confidence helix or low confidence
Yeah.
beta sheets to the linker and give this more flexibility.
Yeah.
And you can just tune it up a little bit, refine more or refine less.
Yeah, it's not automated. You'd at this point you'd have to, as a user, I think I need to change this part or something. Okay, so Jeremy?
Okay. So you basically clone the GitHub. There's a link on the GitHub to the Collab. But so you install everything, which I have done, and upload the PDB. So I will upload the AIP.
So if you think about Bilbo MD, there's all those input files you have to set up.
Not anymore?
No, back in the day.
Back in the days.
Right? So this is where we're at with it. You download the GitHub, it creates these input files, and then there's a separate C program that will just rip through the input file.
You should hire Scott.
I would
We just
totally.
need to add this as a module onto
Yeah, we'll just take him with us.
falling out of a tree.
So yeah, you set your run name, and then there's various options depending on if you have a monomer, a multimer, or if you're using PAE files, or have like fixed distance constraints for like for example sulfide bond predictions. I should probably see the.
So what did you just upload? I missed that. Just, you uploaded a PDB file and a PA?
You can, but.
No, but what did you,
not in this
okay,
case.
you didn't do that just now?
No.
Oh, okay.
I think it's not in the right directory, so I will reinstall the thing.
Yeah, we used to, I told you, we used the Juniper for the first version of ScÅtter.
Yeah.
I mean, remember Scott? The Juniper?
Very not good.
Very not good.
Yeah.
But it was just probably, how was it?
Well, it would, plus you were running it on Google's shared compute.
Yeah.
Yeah.
Sometimes get this GPU, sometimes get that one.
Yeah.
Every time you did it, you had to like reinstall fucking everything, PyTorch and all that. It'd take
Yeah.
like 20 minutes to set it up, and then you'd upload a file and it wouldn't work, and you'd have to. Anyways, if you have your own, I mean, these notebooks can be stable if you're running on your own servers.
Yeah.
At least my experience, I don't particularly like this. I mean, they're nice for dev work, that's for sure.
Yeah.
For production.
Do you still do some SAxS?
No, I use old data. I try to find many ways to republish it.
Is the carbon R, is that GitHub repo?
I do recognize Smart California.
It'll
Yeah,
be
P4,
interesting
P6.
if you send it to Brant and see what he thinks.
Oh, I did.
Oh, the Smart Cal stuff?
Yeah.
And what do you think?
Yeah, he wants to follow up with it.
Yeah. Provides a testable hypothesis.
Yeah, but I think getting it right with users is the big problem for us right now. The Jupyter stuff is, I think you have to be somewhat proficient problem solving, but we'll get there.
Just use that.
Vibe code it out, man, you'll be fine.
So you're speeding up the computational part for the SAxS because it's coarse grain. I like the idea of Ramachandran sampling, like the linkers, I really like it. It's kind of very fast probably.
Yeah.
Super fast.
Yeah. And then it's just that SAxS discrepancy that you're minimizing.
And there's a geometric penalties as well.
Do you want to show your slides?
Yeah, well.
Well, the other great thing is you're not dependent on the MSA.
On well,
Well, you
the
kind of
sequence
are.
alignment comes out of the secondary structure.
Yeah,
If you're
not-
relying on an
not as
AlphaFold
to-
structure, you are.
So we have like standard geometric penalties for like neighboring the alphas and non-neighboring the alphas. And then distance restraints, which can be like user input. DI0, that's the ideal distance. And W0, omega 0 controls how strict the penalty is. So lower value is more strict. And then this is a little bit more complicated. Could I use a board maybe?
Absolutely.
I don't really understand that.
You can find a pen that works.
Yeah.
So if I could draw like a protein, so you have a helix, a link or something, and then maybe some sheets, and then another linker. So first we take the endpoints of the helices and sheets, and then we just replace them with a segment. And then the linker simplification is a bit more complicated. So we'll take like 3 points on the linker and construct a triangle. If there's nothing going into the triangle, then we can remove the middle point, and we can do that across the whole linker. So like this one here will just be represented by a segment. If we have like a linker, suppose like this, and then there's like something going through it, then the middle point will be retained. So this SKMT representation provides the simplest number of points that preserves the overall entanglement of the protein.
I have a question. In this case,
Yeah. You can see.
the classical regions confirmation are disrupted and are more simplified, right?
Sorry, what?
Flexible. Say it again.
Classical regions, like no structure.
Yeah.
Regions, their shape or their confirmational accuracy are oversimplified. We now consider anything about it.
So in this Rive calculation, they're simplified, but in the search space thing, they're not, right? So what he's trying to, if you have a random chain, right?
Yeah.
The question is, when we're resampling the structure, is how protein-like is it, right?
Yeah.
And they, what he's explaining here is this calculation called the Rive. So they looked at a lot of protein structures, calculated the Rive for, I don't know, how many were in there?
10,000, I think.
Yeah, 10,000. Right?
I have another slide on that.
Yeah, it's the next slide. But there's an interesting observation that that kind of bounds the Rive, right? And we can, as we're doing the search, right, and we're calculating its SAxS data, we also have to try and make sure that whatever is the SAxS discrepancy isn't the only thing that we're interested in. We're also trying to balance it out against is this protein-like, right? And that's this simplification that is showing with the Rive. So go ahead.
Yeah, so this just simplifies the calculation, provides basically a piecewise linear segment of the protein. And then we calculate the average crossing number, which is given by that formula. So the omega ij is just the crossing contribution of edges between xi and between two edges. So right, so if you see on the image, if a line goes over another one, it's +1, and the another one is a -1. But here we don't care about the signs. So we basically just sum up all the crossing contributions across all orientations. So so the number of edges crossing over another edge depends on how you look at it. But we sum over all of the orientations. And then we just penalize that crossing number based on the lower bound, which I'll show on the next slide.
Right. So if you take a protein shape, right, and you look at it one way, you'll see things crossing. And then you turn it, crossings will change. You do this integrate over all perspectives. You'll calculate it, this overall crossing number. So go ahead. This is the best part here.
But this formula was derived from the Gauss linking number, which is another well-known result for Rive in knot theory. So you can see the SKMT representation on the left, and the right shows you the like the bounds on ACN we saw in like crystallographic data of like 10,000 proteins. So the bottom axis is the SKMT length, so like the number of segments we have in this representation.
And so for the ones that kind of fell below the bound, if you look at the structures, right, the electron density was like poorly defined, and you had to kind of guess at the chain trace. So you can kind of see, you can kind of detect, I think, misbuilt models. But anyways, keep going.
Are these the high resolution structures from the Richardsons, or is this?
No, this is completely different.
Oh, okay.
So that's that for the geometric stuff.
Yeah.
That was what you talked about with SmartCal.
Do you recognize that, Nick? Do you want to try the demo now?
So before you go, it's okay. What's the final solution here? Which one was the right one? It's on a chi-square, I think this one.
No, I don't.
It's okay.
This
It's
is
not
principal component analysis,
Yes.
right? Yeah.
But anyway, this one was just the final C one with carbonara 1.77. Anyway, that's just the black one right here.
Yeah. Single one. The best single one, right? That was.
So it's good to assume that this kind of approach is good for when AlphaFold completely non-confident.
No, it's when you're not confident in AlphaFold.
So are you using this Noether stuff to restrain your repacking algorithm?
Yeah,
So
yeah.
that you don't get sort of non-proteinaceous shapes, or what? How are you using it in is it in Clinks or is it in carbonara?
This is in carbonara. Yeah, no, we were thinking about, as you kind of resample spaghetti, right?
Yeah.
There's a how can we kind of keep it within some kind of bound? What's protein like?
So you have a mathematical function that will penalize you if you get into too unfolded or too
It
folded.
would be too unfold.
Wouldn't be the other way around, like too many crosses, unrealistic knots or something.
That was the upper.
That's the upper. Yeah. So it'll penalize on either side. It sort of will restrain it to that band.
Well, there's certain chain length, right? And you can see there's a lower and upper bound to that chain length.
The x-axis was chain length, the number of segments.
No, why?
The y-axis was chain length.
So x-axis was the number of SKMT segments.
Within a particular protein.
Yeah.
And then the y-axis was
The rise.
The ACN.
the number of crosses.
Yeah.
This is why we don't like Jupyter notebooks.
Yes. No, I agree.
Half an hour later, it's still installing PyTorch or whatever. And then fails for some unknown reason, and you have to start over.
While this is going on, what are you thinking about in terms of dealing with ensembles in your SAxS?
Right, so that's the next logical place to go. There's Carbonara 2, which we're working on, which does parallel runs to try and deal with the ensembles. You can do complexes too, but it's more, you have to define the interfaces that you call rigid, like you were showing yesterday. And then that's maintained, and the rest is kind of same.
It was working earlier.
The MD is still very important at the end. Right?
Well, that's true for AlphaFold too, right?
I don't.
They
do
have
like
the
a
option.
relaxation stuff.
Yeah, you can run and relax at the end. You don't have to.
And how trivial is this to add, something like footprinting data or other things? Is that what you guys are thinking also?
So that's his PhD is on adding that as a potential inside the carbonara fund. Yeah.
What about rotamer outliers? I guess this has nothing to do with rotamer
No.
outliers.
Yeah, so the clinks thing would potentially have the rotamer outliers, but I mean, that's, the clinks was, the value of it was, or is that we're only looking at amino acid conformations that have ever been observed at high resolution. Right? So I'm not relying on MD, because with MD, I always end up, when you're doing a simulated annealing, you'll have unrealistic conformations, right?
Yeah.
And then you find out at the very end after 2 hours or something.
But it's in what, 1 in 3,000? No, I forgot what the prevalence of outliers are. No, 3 in 1,000 residues will be a true outlier according to Jane Richardson. So it's in your data.
Probably. But it's, SAxS isn't at that resolution.
You might investigate containerizing this. Put it into a
Yeah,
Docker
no.
container. You don't have to do this once, or whenever.
What is stopping this from being applied to, say, membrane proteins? I guess you can't do SAxS for membrane proteins too much, but I guess since you're not relying on AlphaFold as much, it might be better. You have to have like a membrane protein flag on it.
Well, we need a way to model the membrane, right? That would be the challenge at that point.
I mean, you're not modeling the water here either.
So the form factors, all the amino acids, those form factors have been approximated.
I see.
Yeah. So for every residue, we have the corresponding form factor, which includes hydration.
But in terms of this knot theory and all that stuff, and connectivity and secondary structure, right? It's not. There's no water there. That's just all sort of learned. And same within a membrane protein is kind of what the membrane does to protein is learned.
Yeah, the bounds are observed limits, right? So in the same sense, would it be different for a membrane protein? I have no idea.
I mean, it's like a flag. You're not to switch. Like this is in solvent and this is in membrane.
Yeah.
I think that would be the major identifying thing. You'd have to develop a parameter set. Anyway.
Yeah. That would be for the next student.
So maybe this is for the student after that. So you go, see what you can do with footprinting, right? Is it ultimately the goal to have multiple different types of experiments that are all going to feed in and give you constraints?
Yeah, I mean, this is purely about modeling the solution state. So, SAxS, the footprinting data, potentially neutron scattering. Yeah. And then
NMR.
Yeah, NMR. A little more complicated. Yeah, so it's you want to have a model that's with high confidence agrees with your set of observations, right? So
I guess my question is once you have the footprint, where can you use both SAxS and footprinting constraints at the same time?
yeah, that's the goal. Right? Yeah. So you'll get that residue specific information from the footprinting, and then overall topology from the SAxS, and then come out with some kind of refinement to validate. Right? Yeah. Okay, doesn't look like it's going to work for you. We're here all week.
Yeah. You're gonna have to show us.
He's just talking about this one demonstration, not the rest of your project.
Yeah. That's right. We'll have to talk to Chris.
User friendly.
That's the hard part.
Yeah. Well, I told you, hire Scott.
Well, collaborate.
Like fish and chips, you wake up.
The greasier the better.
Well, yeah, should we close it here?
Yeah.
Should we give them a
Yeah.
Thank you guys.
Well, I think if we could get it into part of your package.
I think so.
Okay.
It's really, it's just,
We're waiting for that
like you're
demo.
saying, you're just making a bunch of
Imagine ending it